Hello everyone, today, it's my pleasure to share with you a new study in the field of inflammatory bowel disease, oral proton-reprogrammed nanomedicine to break the inflammatory bowel disease Clostridium difficile infection vicious cycle. Clostridium difficile infection, CDI, is a major complication of inflammatory bowel disease, IBD, but the complicated treatment strategy of antimicrobial drugs and immunosuppressive drugs cannot effectively control the syndrome of IBD and CDI. Glycolysis in intestinal epithelial cells, IECs, leads to excessive production of lactic acid, which is considered to be a key factor in the destruction of the intestinal barrier by toxin B of Clostridium difficile, TcdB. This study revealed that UCP2 is highly expressed in the intestinal epithelial cells of IBD patients, leading to mitochondrial dysfunction and metabolic reprogramming toward glycolysis. This resulted in lactate accumulation and cellular acidification, which significantly enhanced the toxicity of TcdB. To break the vicious cycle of IBD-CDI, we developed an orally administered proton reprogramming nanodrug, OPR, which effectively alleviated symptoms of both IBD and concurrent CDI. Analysis of human transcriptome data and pathological examination revealed that UCP2 is significantly upregulated in the intestinal epithelial cells of IBD patients, showing a positive correlation with disease severity. The abnormal elevation and activation of UCP2 lead to acidification of intestinal epithelial cells, enhance TcdB toxicity, and disrupt the intestinal barrier. This study designed and synthesized a UCP2 targeting nanoparticle, named OPR, which employed genapin, GP, a UCP2 inhibitor, as the primary active component. Through structural modification, OPR can stably pass through gastric acid and achieve sustained release of genapin upon reaching the intestines. With a negatively charged surface, OPR can specifically target the positively charged inflamed regions in the colon of IBD. In an IBD mouse model, mice in the OPR group exhibited significant body weight recovery, the lowest DAI scores, and marked improvement in histopathological damage scores. Compared to first-line IBD therapeutic agents such as 5-aminosalicylic acid and infliximab, OPR demonstrated superior efficacy. OPR significantly modulates the gut microbiota by enhancing alpha/beta diversity and reducing the abundance of Clostridioides difficile. Potential mechanisms of OPR against C. difficile toxin include inhibiting RAC1 glycosylation to protect the cytoskeleton, reversing cellular acidification, and restoring cellular ATP synthesis. In IBD-CDI mouse model, OPR demonstrated superior efficacy compared to the combination therapy of fidaxomicin and infliximab. The therapeutic effect of OPR against CDI does not rely on direct bactericidal activity but rather on restoring and enhancing the intrinsic toxin resistance of intestinal epithelial cells. OPR restores mitochondrial function in IECs by inhibiting UCP2. Additionally, OPR reduces mitochondrial DNA release by blocking pyroptosis in IECs. Furthermore, OPR restores the levels of various intestinal tight junction proteins and repairs the intestinal barrier. OPR reduces the release of mtDNA from IECs, inhibits the cGAS-STING pathway in macrophages. OPR decreases the expression of STING, pp65, and PIRF3, downregulates the levels of TNF-alpha, IL-1-beta, and IL-6. Analysis of human intestinal single-cell sequencing datasets revealed that UCP2, STING and GSDME are upregulated in intestinal of IBD patients. Mouse intestinal transcriptomic sequencing results identified a substantial number of differentially expressed genes, DEGs, between the OPR and IBDs. These DEGs were enriched in pathways related to glycolysis, pyroptosis, STING signaling, and tight junction proteins. This study reveals that UCP2 is highly expressed in IECs of IBD, and reduces the resistance of IECs to TcdB. This study develops an oral proton reprogramming nanomedicine, OPR, that can alleviate IBD and CDI syndromes. This study reveals that metabolic reprogramming of IEs represents a potential therapeutic target for IBD-CDI.